is Kyla Belshner, and welcome back to our second episode of our Symbiotic podcast series. So, as our guest today, we have Mia Benami, who did a summer-long internship at A2 Biotherapeutics, and so we're going to talk a little bit about her internship. So, Mia, can you just tell us a little background about your internship and your experience?
Sure. So this summer at A2 Biotherapeutics, I worked with the Discovery Research Department in the molecular biology team. So it was a lot of lab work, and they basically just integrated me into their workflow. Anything they did molecular biology-wise, I then helped out with, and I had a mentor, and he just basically taught me the ropes, taught me everything I needed to know in order to then help them out.
Very nice. Okay, so how did you come upon an opportunity like this?
So at Pierce College, we have a program, a biotech program called Aspire. And this program basically encompasses everything having to do with biotech, but it also offers courses. And right now it's expanding. But when I took the courses, it was Biotech 1 and Biotech 2. And these courses basically prepare you for industry. And once you get to the second course, you work on your own project. Now, after I finished this course, I was looking for internships, looked into REUs, things like that. But the director of the program, Dr. Kamajaya, he approached me and said they had this grant going called the NSF Excellent Grant. And this grant basically gives students internships, and they pay for the internships so that the companies they work with don't have to pay for the internships. And what you do is you just write an essay and you get matched up with a company. Now you then interview with the company, and if it's a good match, then you get hired. So I came upon that application and I applied for it, and I ended up matching with A2 Bio, and from there it ended up being perfect. It was a great opportunity.
Okay, so I heard you mention REUs, and I know that REUs are research experience for undergrads, and I know a lot of students have heard about those. So what's the difference between an REU and an internship like you just did?
So REUs are also funded by NSF, but they take place in on college campuses. So a college will put out onto a portal that they're looking for interns for a summer research project that they have going on, and what you do is you apply, and they're very competitive. They look amazing on a resume, but the field becomes very convoluted because there's X amount of RUs available every summer, and there's probably 5 times the amount of students looking for internships. So you're kind of testing your luck when it comes to RUs, and the more established and like Ivy League schools like Yale or Cornell who put out REUs, their applications will be really, really hard to get into. So the main difference is that this internship is much more accessible. And as a community college student, it's a lot harder to find lab experience. REUs are great, but also you're again at a disadvantage as a community college student because you're coming from a lesser known college. Versus the people who are applying to these RUs who go to UCLA, let's just say, are coming from a more reputable college. So I did apply to RUs, but I found myself leaning towards this experience, not only because I was actually getting paid more for a 10-week program, but it was close to home, so I didn't have to travel, and it was just more accessible. I found myself feeling a lot better with this opportunity versus having to go and go to a whole different state for a couple of weeks, live in a dorm, things like that. Depending on the program, some programs don't even pay for your living situation when you go abroad. So I think that an opportunity like this is just a lot more accessible to community college students.
Okay, so would you feel like you would recommend this for other students? Do you think that it benefited you in a lot of ways?
I 100% would recommend it to other students. I mean, I think everything I learned this summer are things that I'm going to just take with me for the rest of my life. I'm actually a pre-med student, so my end goal is not biotech per se, but everything I learned during this internship could be applied to other fields of science, which I felt was like something that a lot of students are missing. They're missing just overall experiences that make their resume look better, make them look more like a suitable candidate when transferring. Community college students, we're at a disadvantage, as I said before. It's much harder for us to get these experiences. So when you come across an experience like this that gives you so much knowledge in the lab and gets you networking and things like that, I think that's invaluable. So I would recommend this to everybody who asks.
Okay, and say there was a student that wanted to do an internship just like you did. What's one piece of advice that you would give that student who's trying to get an internship, especially at the community college level?
I think I would say that, be persistent and don't say no to opportunities that feel lesser to you. I had the RUs coming one way and I had this coming the other way. And RUs definitely look better in the college perspective, I want to say, on a resume or on an application. But the one thing that they don't have that the opportunity that I got does have is the amount of networking I got to build. Agora, Pierce College is in Woodland Hills. Agora is about 15 to 20 minutes away. The network that I built at A2 Biotherapeutics is a network that will follow me through my career. I made over 30 LinkedIn connections just from working over the summer for 10 weeks. So I would say be persistent. Don't give up. These opportunities come, in the most least expected times. This opportunity actually came because my Biotech 2 professor worked at A2 Biotherapeutics and put in a recommendation for me to internship there. But I would say just stick to it because the opportunities will come, and don't feel like as a community college student that you're less advantaged because you make your own advantages.
Okay, so that makes sense. And I heard you mention networking. Can you go into more detail about what that is and why is it so important, especially as community college students and who are trying to either go pre-med route or go into the biotech industry route?
Sure. So networking is very important, especially when it comes to later on getting jobs, making connections, things like that. Networking is basically just building up people that you know in the industry that can help you later on, getting connections. LinkedIn, if you don't have one already, you have to make one. LinkedIn is basically the holy grail of networking. Anybody who have linked on LinkedIn then becomes a connection that you can use later on. I was actually talking to somebody at A2 Biotherapeutics. He's higher up in the company. And he was mentioning that when he was going to hire people, if that person didn't have at least 5 LinkedIn connections that he knew of, he would not hire them because he didn't know anything about the person. He didn't know if that person was somebody that he could trust based on their LinkedIn connections. So networking is key to being successful in these industries. Med school, biotech, getting into a lab. If a professor sees that you maybe worked with somebody they know at a different company and you're now a LinkedIn connection with them, that makes you more valuable than somebody who looks good on paper but has no connections to the professor. Not just LinkedIn, but also just getting to know your professors. As community college students, that's a big advantage to us because our class sizes are much smaller. We get to be able to talk to professors, get to know them on a more personal level. So keeping those connections and building them now early on in your career is really important.
Okay, so did you feel like you were prepared for when you went into this internship? I know that you mentioned how you took this biotechnology program at Pierce. Did you feel like you had enough lab experience, any bio or chemistry background that you needed? Did you feel like you were prepared when you were stepping into the lab and ready to intern for them?
The biotech program definitely gave me a lot of knowledge on how to work in a lab. But I will say that I had no bio experience coming into this internship. All the bio knowledge I had, which was very little, came from the biotech program I had taken, because I had never taken a college course bio class. So I was a little nervous going into a molecular biology team having no bio in my, arsenal. But another great thing about having an excellent internship like this is that they taught me everything I needed to know. There was no, oh, you don't know this? That's not acceptable. There was just, okay, let's go over the basics. Let me teach you that. Let me give you the background on that so that you can succeed. I really, really feel like that was the highlight of this internship. I learned so much from this internship without having to feel stressed or nervous that I didn't know enough coming into it. With REUs, that's another thing. Like, sometimes with REUs, they expect you to know a little bit more than surface level. So that when you apply and they see that you have no biology and you're applying to something revolving around biology, they might take a second glance and be like, well, we don't think you're a good fit for this. Even though they say that this is supposed to be, a starting experience, something that'll give you more lab experience. But with this NSF Excellent grant, that wasn't the case. We really came in not having to know much other than, basic lab skills, being able to be adaptable, flexible, and being able to learn and catch on quickly, I would say.
Okay, very nice. And what was the hardest challenge you feel like you had to overcome during this internship?
I think for myself, I'm a little bit introverted, and going into a new environment felt like a little bit of a toll, took a little bit of a toll on me at first, because I was scared that I didn't know anybody, and I was having to learn all this new stuff and having to learn all the lab regulations and rules that A2 Biotherapeutics demanded, and things like that. And I think that learning how to be a little bit more extroverted and learning how to just, overcome my anxiety and things like that was the hardest part of this internship. Luckily, there was another intern who came from Cal Poly who was part of the CERN program there. We became close friends, and we kind of, navigated those waters together. So I would say that would be the hardest challenge I had. But another great thing about A2 Biotherapeutics is that every single employee there, every single colleague there was just absolutely amazing. Nobody ever made you feel unwanted or not cared for. And the front desk lady, she was absolutely amazing. My mentor was absolutely amazing. He's kind of like a father figure now. He was always so nice and always making sure that I had everything I needed. And you come into an internship like this, and they set you up with a desk, and you're in the bullpen, and all the people around you are geniuses, and you're hearing all this biology talk around you, and you're I don't even understand half of the stuff you're saying, but oh my god, you sound amazing. And the person behind you is a senior in the company, and it was insane. And you all of a sudden have 2 monitors set up that you can look at data through, and the lab is literally 3 steps away from you, and people are gowning and gloving right in your surface area. It's insane. So I just really feel after I got over that challenge and hurdle, it just became so much fun.
That makes sense, especially for a lab being that large. It can seem hard. But did you feel like a fish out of water there, or did they feel like they were accommodating you and you felt like you were where you belonged?
Yeah, so accommodating. Honestly, at first I came in first day and I was oh my god, I don't know how I'm gonna do this. These people seem like geniuses, so intense. But then you start talking to them, and they're all so lovely and friendly, and every single person there, like you're in all staff meetings and everybody's smiling, and then you walk into like discovery team research weekly meetings and everybody is super chill and they have a good vibe going on, and you know, they're asking how everybody's day is, and they bring in bagels every Wednesday, and you have, all of a sudden you have an event where they're bringing coffee for everybody and everybody's chatting, and you know, it was very welcoming. And I think A2 Biotherapeutics is considered a startup, but I'd say they're a little bit bigger. I think they have around 70 employees in 3 different buildings, and I would say every single one of them is just so nice and so welcoming, and I think that's rare, which when you go to an REU, as I was mentioning before, you know, I don't think you meet as many people as you would if you work directly at a company. You know, in an REU, you probably have your PI, and then you have a couple other interns and maybe some grad students. But here you're really building connections with industry people.
Okay, so now that we've talked a little bit about the social aspects and the actual internship process, can we go ahead and circle back to A2 Biotherapeutics, and what do they actually do? What does their lab do?
So A2 Biotherapeutics is a CAR T cell therapy research. So basically what they do is they try to target cancerous tumors without killing healthy cells. And what makes them special is that they're actually targeting solid tumors. So if you think about chemotherapy, what chemotherapy does is it targets all cells, including cancerous as well as healthy cells. So that's why people have such severe side effects from chemotherapy, because they can't really regulate what the chemotherapy is killing. It's just killing everything so that you can get rid of the cancer and then hopefully get better after that once the cancer is gone. What A2 Biotherapeutics is trying to do is they're trying to target just the cancerous cells, so that your healthy cells will not have any issues and you will have a lot less side effects from it. They have a thing called TMOD. And TMOD, what it does is it has 2 receptors. It has a receptor that binds to cancerous cells, but then the other receptor it has a stop receptor. So what that receptor does is it stops the TMOD from killing healthy cells. So it'll bind to cancerous cells, but the cancerous cells don't have that receptor that binds to the stop receptor on the TMOD. While your healthy cells have not only the killing receptor, but it also has the stop receptor. So when that stop receptor attaches onto your healthy cells, it tells it to stop the killing process.
Wow, that's really cool. So did you help with this?
So when you're in discovery research, a lot of what you're doing is very experimental. They do have things going on in the discovery research department that's helping directly with their final product. But what I did a lot of the time is I helped deal with a lot of the DNA, RNA, anything they needed to then go on and help with T cell growing, killing assays, things like that. And I also helped with automation in the lab. They are very automation forward at A2 Biotherapeutics, which was amazing. They have a lot of really innovative things going on in the lab, which was great.
Very nice. So I do know that your biotech project was also automation based, I believe. Did you do anything similar? Like, what was your biotech project about?
Sure. So my biotech project dealt with a machine called the Opentrons, which is an automated liquid handler. And what I did is there's a project called Tiny Earth, which was originally started by Johns Hopkins. And it's a project that has community college students trying to find antibiotics from soil samples. And part of that project is going from a master plate of bacteria onto plates swatched with antibiotics that you think you've found from your soil samples. Now, this process is basically picking and patching. So you're picking from your master plate and you're patching it onto a grid on your, then now what you're considering antibiotic plates and seeing if they grow and if what you found is actually an antibiotic. This process is very tedious. It takes a lot of time, a lot of effort, a lot of precision. You got to really stay focused when doing it. So I wanted to be able to automate it so that students wouldn't have to worry about that portion of the experiment, and they could go on and do whatever else they needed to do in the lab. So I used the Opentrons to automate a pick and patch protocol for the Tiny Earth project. And I was successful using square Petri dishes, and I am now going on to drafting a research paper about it. But how this tied into A2 is A2 also uses the Opentrons for certain experimentation in the lab, including killing assays and things like that, serial dilutions. But above that, the Opentrons is probably the cheapest version. The bottom level of automation that they have in the lab. They have another thing called the Bravo. The Bravo is kind of like the Opentrons, but it's a lot more precise and complex. I saw it working there, and it was really cool. But the one advantage of the Opentrons over the Bravos, the Opentrons is more modular. So you can add in things like the thermocycler module, things like that. And, Kyla has worked on the Opentrons, so she knows that it's very versatile. We both have 3D printed things for the Opentrons that we can then click in because of the deck, and it becomes very accessible for a variety of things. But another piece of automation that I worked on there is called the Hamilton Starlet. And the Hamilton Starlet is like a one-stop shop for automation. It basically uses a variety of different docking stations, including like Epic cartridge docks, 96 well plate docks. You could put like a cold block on there. And I automated a Golden Gate assembly process, which is like a one-pot mixture for DNA, where you can then PCR your product. Wow. It was really cool. And we can go into Golden Gate assembly later on. It's a little bit of a complex topic, but it's a pretty new method that people have been using. I think it's 2000 was when it was first introduced. Don't quote me on that.
I won't.
But yeah, so I was able to use the Hamilton Starlet, which was absolutely amazing because I was very limited to Opentrons automation. And they even had automated pipetting system. It was actually semi-automated. So what it did is you have this machine with a handle to it, and you just click a button and you put it into your liquid, and it vacuums it up, and then you transfer it over. It's on a dock, and you push it down, and then you click another button and it pushes the liquid out. Wow. So yeah, that helped when you had 96 samples that you were trying to use, and inoculations and things like that. It was really helpful. Yeah, so automation was very prominent at A2, and I really appreciated the exposure to it.
So when it comes to a lab like A2, why is automation such a big deal? Why would someone spend all this money on all these different robots when you can just get someone to do it?
Sure. So automation is mostly about efficiency. Automation, how people like to describe it, is it saves you human time. Now, real-time wise, the robot can probably do it at the same speed as you can for most situations. There are situations where the robot can do it faster, or it can even do it slower. But what it's saving you is human time. You can step away from the robot the second that protocol starts, and you can go do the probably millions of other things you have to get done that day. When you're in a lab, it's usually you're working on 3 different experiments at the same time. It's very fast paced. You're trying to figure things out. You're troubleshooting something while running another experiment. So automation really just is more efficient. It saves you a lot of time, human time, not real time. and it's more precise. A robot can probably do it a lot better than a human can, based on results we get from robots. Things that are prone to human error, a robot can do without the human error to it. There's also other sources of automation. They have this amazing Q20 freezer that just catalogs all their components, their primers, things like that they need stored and logged, so that you go through Benchling, you insert what you want to get out, it tells you how much of it is left, you put it into the queue, and then the robot a couple minutes later tells you it's ready. You go over to the robot, you click a button, and the robot has whatever primers or constructs you need for you at the click of a button at that point.
So I know at A2 there are multiple departments, and we've been going over a lot of the research and discovery department. What other departments do they have, and what exactly do they do for A2 Bio?
Sure. So basically, A2 Bio is like a big wheel. You start at the discovery research, you move on to process development, then process development then moves on to manufacturing once they've figured out that everything is safe to then go on to manufacturing. Manufacturing puts out the product. Now, when they want anything tweaked, when anything needs to be changed, anything like that, it goes back to process development, and then process development goes back to discovery research, and we basically just continuous contact with all the departments. So while you're only in discovery research, or that's where you're working, you're very integrated with the other departments when it comes to like workflow. So process development, what it does is once discovery research has come up with a good product and they think it's something that's feasible and safe and it works, and they think it can continue on to the next stage, they transfer it over to process development. Process development, basically what they do is they figure out a way to give this to a patient in a safe way. Whether that's, making sure that everything's sterile, making sure that the product can be developed in a safe way, things like that. Because discovery research, their one goal is to find your final product, to find, what can TMOD then connect onto. Do both the receptors work? What needs to be changed in the receptors? Things like that. So process development then finds a way to make this feasible in a patient. And A2 Biotherapeutics product is not actually like the patient comes in, gets something. Our product is blood. Blood gets sent to A2 Biotherapeutics. A2 Biotherapeutics takes the cells from the bloods, they put in TMOD, and then the blood gets transferred back to whatever clinic or hospital that patient came from. And then that hospital probably works with A2 closely. And then the patient is given back their, cells plus the CAR T cells that are now in the blood. So process development makes sure that everything will run efficiently from start to finish, and manufacturing gives out the final product after process development has made sure everything is safe. So process development has its own laboratory. Actually, process development and manufacturing have their own laboratory. Process development has a mock laboratory of what manufacturing does. So when you're dealing with blood, biosafety level has to be very high. So they have clean rooms where people have to gown and glove. You can't be wearing the same shoes you wore to work. You have your own shoes that you have to put on. You're fully gowned. Everything is protected because blood also has airborne diseases that people can get. So not only does the scientist need to be protected, but the person who is then getting that blood needs to be protected. So yeah, all the departments work pretty seamlessly, and I got to tour the other buildings and see the other laboratories, and it's really amazing because at A2 Bio, all the laboratories have this amazing just gallery window where you can walk with the people inside the room, which is really rare actually for a lot of laboratories, where you can truly see inside without compromising what's going on in there.
So earlier you said that you were a pre-med student. So
Yes.
how would you take what you learned from this experience and apply it to your later on life, whether that's in medical school, residency, or just in general?
So a lot of pre-med students focus on getting, all the medical, internships or work experience at hospitals, volunteering, becoming EMTs, medical assistants, things like that. But a lot of people don't realize that research experience looks just as good on a college application. Of course, you still need to do, the volunteering at hospitals and getting in that medical shadowing, things like that. But this research experience is directly tied to the medical industry. Everything we're doing, CAR T-cell therapy is a medical industry. So, whether you're helping a patient directly or indirectly, which is what A2 Bio is doing, you're still within that medical field. And I personally love lab work and I love being in the lab, and I wanted to get a lot of that done before going into, med school and things like that, which then residency, you go into research as well. So having this in your back pocket helps you a lot, because medical research just gives you the other side of the medical field that you don't really always consider when going into the field itself.
So I am aware that you were originally debating whether going to medical school was right for you. I know it's a very long journey, versus working in a lab. What did this experience do for that? Are you still considering both options, or are you driven to one or the other?
So I think the main thing I realized about this job during this internship is that I didn't like the 9 to 5. I didn't like having to go in, every day, sit at a desk. You're in the lab a lot, but there's also a lot of desk work to it. And I think I found myself a little bored. It's a little repetitive. I think, especially in discovery research, I will not say this applies to like all of biotech or all lab jobs, but I felt like in discovery research, you're redoing experiments all the time. It's trial and error. You're not going to always succeed, things like that. It does become a little like, do it again. Oh, come in tomorrow and do it again. Mm-hmm. Which I didn't have an issue with. My main thing was when I decided that I wanted to go to medical school, it was because I wanted to feel like I was having an impact on somebody else, to feel like I was helping out. And you're very disconnected from, the patient when it comes to research, especially medical research. You're never really seeing the outcomes unless, it's shared to you by the company. And so I think the one time I was really like, oh my God, something I did was actually used, was they basically test on mice. And we went to the mice facility, me and Daniela, the other intern, and we got to see a mouse injected with RNA I had made, which was really insane, because I was like, oh my God, it's actually being used for something. And yeah, that was like a oh my God moment, like, wow, it's actually being used for something. But I did feel like the repetitive 9 to 5 just wasn't something I wanted to do. But nonetheless, I will, if somebody told me, if you could go back, would you not do the internship? I would say absolutely not. I would do it no matter what, because you need this experience. You need something like that to help you realize, what's for you. So if you are thinking of going into the biotech field and you are thinking, this is it, this is what I want to do, go do an internship. Go get your hands dirty in the field so that you can truly, truly know that that's what you want to do. I have shadow doctors, I have done the lab work now, and I'm still not 100%. I'm still doubting the decision. I'm still but what if, you know? But I think that, that's life. Yeah. And I think that no matter what, I'm always going to have, that little voice in the back of my head being oh my God, it's 15 years. What are you going to do? You're going to go into student debt. This is going to be an insane process. But I think that, ultimately, it's really just, believe in yourself. believe that you can do it. Believe that your experiences have taught you what you need to know for the future. And I think that this was a really important experience because it taught me that maybe I don't think I can work the 9 to 5.
Well, thank you so much, Mia, for answering all of our questions. I really appreciated having you on this podcast. And we are going to have another episode out soon, so stay tuned for that. Thank you, guys.
Thanks for having me.